Hello everyone. For my VoiceThread Project 1, I'll be reviewing the 2021 article by Carbonaro et al. Named Gene Therapy Using AAV8 in Vivo for Disease Stabilization and a Bimodal Gene Therapy Approach for the Treatment of ADA-deficient SCID. Adenosine deaminase, or ADA deficiency, is an inborn error of metabolism affecting multiple systems and causing severe combined immunodeficiency, or SCID. SCID, also known as bubble baby disease, is recognized by the medical community as a pediatric emergency. SCID was the first disease that was treated via bone marrow transplantation from compatible, healthy donors like siblings or parents. This genetic mutation leaves the child without a functioning immune system. SCID babies have a profound vulnerability to fatal infections when they encounter normal germs and bacteria from the surrounding environment. This disease was depicted in a Hollywood movie starring John Travolta called The Boy in the Plastic Bubble, coining SCID patients as bubble babies. Babies with SCID seem to be healthy at early stages, up to 5 months old. The first symptoms can include the flu, being lethargic, peripheral cyanosis, and not eating or sleeping properly. When this disorder goes untreated or undetected, it can lead to the death of the baby before they turn 1 year old. The lifespan of SCID patients that are treated with bone marrow transplants and gene therapy techniques are considerably long when compared to untreated patients. Children need to have blood tests and skin biopsies for the diagnosis of SCID when they're born, but non-invasive prenatal testing can be conducted around 10 to 22 weeks of pregnancy and allow parents to know the genetic makeup of their child, to note if they are affected or carriers of any disease. Catching SCID prior to the onset of infection is very important because the 5-year survival rate for those patients is 80 to 95%, compared to less than 3.5 months for those who are not screened for the disease. To start, I would like to give a background on the previous therapies for SCID patients. Unfortunately, patients and their families have to isolate themselves their whole life because of the potential of catching a fatal illness. If the child is not screened for SCID, the baby will typically develop a severe illness that kills them before the age of 2. This truly puts an emphasis on why treatment for SCID was necessary and how it would help patients and their families drastically. For all families, newborn genetic screening is very important because early detection plays a large role in the ability for a patient to be treated. SCID can be curable by transplantation of bone marrow from compatible donors. With early diagnosis and gene therapy through bone marrow transplant techniques, children with SCID have the ability to live longer and healthier lives. Current treatments that are used to keep the child healthy include antibiotics, antifungals, and immunoglobulin replacement therapy. These medications give protection against infection and improve the odds of successful treatments. Initial therapies consisted of isolating the child through screening them out in a bubble to shield them away from germs and bacteria. Bubbles are no longer needed, but isolation is vital before and after transplantation to ensure safety. Transplantation is through non-invasive IVs, and gene therapy techniques are used to edit the infant's immune system with a compatible donor from the family. This allows researchers to develop treatments that create healthy immune systems in compromised children. SCID patients ultimately are able to get vaccines, attend large group events, and experience daily activities without the dread of becoming sick with a compromised immune system. Current treatments to combat symptoms include blood transfusions, blood marrow transplants, ventilators, and utilizing life support when sick. This disease is very impactful for families because having children is difficult. Parents that pass on SCID to their children are carriers of the disease and show symptoms of SCID themselves. Parents must be isolated from their child. Previous treatments consisted of bone marrow transplants from unrelated donors, but these were dangerous due to rejection complications like graft versus host disease. The only working long-term treatment for SCID patients is bone marrow transplants that are most beneficial within the first 3 months of the baby being born. Currently, researchers are able to conduct these transplants in utero and can directly influence the embryo. If this does not work, normal bone marrow transplantation can be conducted during initial stages of life. Bone marrow transplantation is done in utero at weeks 16, 17, and 18 of pregnancy, using the unaffected parent as the donor. At weeks 36, a healthy-looking baby is born, but doctors must immediately take blood tests from the umbilical cord and note if the baby initially has a functioning immune system. At the 6 to 8 month mark, Parents should keep a close eye on the child to ensure healthy immune responses to common colds, flus, fevers, and pneumonias. Treated children are able to have a semi-normal life by performing normal tasks throughout their day. Now, with gene editing techniques, stem cells are isolated and cultured, implementing autologous methods by using cells that are derived from the baby's own bone marrow or blood. The isolated stem cells are manipulated to express the correct gene that is required by the immune system. These genetically engineered stem cells are then transplanted back into the patient. The limitations of curing SCID has been identified through the previous 2 decades of research, and novel technology has been able to progress treatment. By improving technology in the lab, treating babies with SCID in utero is now a possibility and can grant these children with normal lives. Medical centers like UCLA implement novel techniques into pilot studies in hopes to commercialize the product. With the help of pharmaceutical companies, these treatments can provide the world with available therapies for curing babies with SCID. Dr. Cohn of UCLA Broad Stem Cell Research Center has successfully developed healthy immune systems in patients by using autologous methods that isolate the patient's own blood marrow stem cells to correct the defective or missing gene. Bone marrow is extracted from the patient and re-engineered using modified AAVs that carry the corrected gene. The stem cells are then placed back into the patient's blood supply through an IV. In the article by Carbonell et al., researchers used intravenous administration to test recombinant AAV2 and AAV8 ADA vector in ADA-deficient neonate and adult mice. This was done for a bimodal approach that included RAAV treatment followed by infusion of lentiviral vector, modified lineage-depleted bone marrow at 8 weeks. All the experiments in this study used ADA knockout mice, where they were either ADA negative/negative or they were ADA deficient phenotypes. For the control, ADA positive/negative mice were used because they do not express full ADA phenotypes. Results were measured based on the ADA activity, vector number count, survival, biodistributional, absolute cell numbers, and immune phenotype. This study did experimentation on neonatal newborn mice that tested 2 vectors, which included elongated factor alpha gene short promoter and lambda retroviral promoter/enhancer vector. Also, neonatal versus adult mice were experimented on to test how MND-ADA AAV8 therapy worked differently between the age groups of 8 to 10 weeks. AAV therapy was researched as a bridge therapy and illustrated positive results in a bimodal treatment plan. ADA-negative mice treated with 1 dose of AAV8 vector therapy and then 30 days of enzyme replacement therapy post-injection. This enzyme replacement therapy was to keep mice alive from lethal pulmonary insufficiencies for 180 days without more treatment. Our AAV vector copy number was highest in liver, lung, and the heart, and were associated with near-normal ADA activity and thymocyte development. The methods used in this paper incorporate several different control groups and provide a model organism with the proper knockout genes for accurate research. Scientists manipulated various dosages to understand the effects of the treatment, but more research is needed in order to make real claims. The key findings in this article is that the results show our AAV8 therapy works well in terms of survival outcomes in ADA-deficient mice when compared to groups with no treatment. In the bridge therapy figure, the p-values show the statistical significance comparing the treated group to the untreated control. Neonatal results show that the survival percentages are affected by varying levels of treatment dosages. Survival percentages and measuring the appropriate dosages are important for future clinical trials to ensure patient safety. Throughout this paper, scientists were able to provide a successful proof-of-concept experiment that collected data on how MND-ADA AAV8 therapy can be used as a potential treatment for SCID. The results showed that our AAV group did better than the untreated group, and that this novel therapeutic approach can be used for other metabolic disorders. Figure 6 shows multiple outliers, proving that the dataset had points that were very different to other results. Researchers in this article explained that more long-term research is necessary before preclinical models in humans. The conclusions in this article are accurate in terms of the in vivo use of RAAV for gene therapy. The researchers used this study to illustrate a proof of concept and did not specifically make any bold claims, especially when using murine models that do not provide enough data to make accurate assessments for humans. This study was conducted specifically for patients who do not have access to PEG-ADA enzyme therapy, and there were no negative reactions to this treatment. Along with this, the study did not provide long-term results because the mice were euthanized after 6 months, but other animal studies have shown beneficial data.